So we have a way of figuring out if genes are linked on the same chromosome or if they're not. Here are the results that Morgan had with his experiments with the fruit flies, and he found out that out of the different offspring between these two parents here, okay, 965 turned out to be just like the heterozygous wild type parent and having those phenotypes, okay, and 944 turned out like the other parent that's homozygous recessive for those mutant traits. And that's what we kind of saw from the from this worksheet over here, how more often we're going to have certain alleles put together in the chromosome, and if they're crossing over or recombinant, then they would be having a less of a chance, less than 25% chance of actually being seen in the gametes. And that's what's seen here. Okay, those recombinant phenotypes, they weren't really seen so much in the offspring. So how do we figure out if genes are linked or not? Okay, we use this recombinant frequency formula, where we're going to take of all of the offspring within the those two parents, we're going to count up how many are recombinant. Okay, you're going to divide it by total offspring and multiply by 100. Now, if whatever we get, the recombinant frequency, or how often we get recombinant phenotypes, if it is less than 50%, then that means we're going to have the genes are going to be linked. They're going to be on the same chromosome. If they are more than 50%, that means they are pretty far apart, and we would think that they are now on separate chromosomes. They are unlinked. So if we were to take a look at this information, we are going to only focus on the recombinant frequency. Okay, and we have here 206. Okay, plus 185, okay, organisms out of, if we were to go ahead and do the total for this, this was 2,300, I believe. And so when we go ahead and multiply that by 100, okay, our recombinant frequency is 17%. So knowing that, okay, those two traits of body color, okay, and wing shape or wing size is going to be very close together. That means they are on the same chromosome. Okay. Now, let's take a look. If chromosomes have different genes on them, let's say this chromosome has information for gene A, okay, has information for gene B, gene C, okay, gene D, and gene E. Okay, there are, let's say, 5 different genes on this one chromosome. What's happening here is between gene A and gene B, they're super close together. Okay, and because of that, there's like, if we were to go ahead and do a crossing over to split them up, there'd be a very small chance of getting that split up because the genes are so close together. Compared to if we were to take a look at A maybe through E, there's so many more spaces where crossing over or can happen. So crossing over can happen a little bit more. There's more of a chance because that the genes, gene A and gene E, are so far apart. So crossing over between A and B is going to be less likely. Okay, less likely. And compared to, let's say, chromosome A and E, where it's going to be more likely because they're farther apart. Okay, so if recombinant frequency is small, like I said, genes are closer together, preventing crossing over. If the recombinant phenotypes are, the frequency is big, then that means the genes are further away and pretty much maybe also on a totally different chromosome. So now we're going to take a look at certain examples with certain word problems. So just to kind of review again, the recombinant frequency will tell us how far apart these genes are on the chromosome. Are they on the same chromosome, or are they very, very far apart and they're on a totally different chromosome? Let's take a look at this, the practice problems that are here. I want to go over number 3. Okay, so it says, imagine a species with 3 loci. Loci is just like the location where they're at on the chromosome. So on the chromosome, there's 3 different locations where there are different genes. Okay, the recombination rate or frequency between locus A and locus B is 35%. Locus B and locus C is 33%. Okay, and between locus A and locus C, we got 2%. So we need to predict the arrangement of these genes in order on the chromosome. So let's pretend here is our chromosome. We're going to, I'm going to write down the distances between them, or the recombinant frequency, or in other words, the distance. So between locus A and locus B, it is 35%. Okay, between locus B and C, it is 33, and then between A and C, it is 2%. Okay, I just kind of am organizing it in that way. I'm going to actually start off with the largest recombinant frequency, knowing that they're going to be pretty far apart compared to all the other genes. So if I just kind of estimate, these percentages actually translate over to something called map units. Okay, so 1% is equal to 1 map unit. So here, 35% recombinant frequency relates to 35 map units. Is basically how far it is. So since they're the furthest from each other, I'm just going to kind of estimate like how far they are, and I'm going to label that this is gene A and this is gene B, and they are 35 map units away from each other, or you can put percent if you want. Okay, now the next thing is between B and C, they're also pretty far from each other. Okay, but they are a lot closer compared to the distance between A and B. So that kind of tells me, okay, between B and C, and then also between A and C are closer to each other. So that must mean that C has to be somewhere in between A and B. But since there is a smaller recombinant frequency between A and C, C's got to be closer to A. Okay? So in this case, I'm going to say here maybe C is over here, and the distance between these two would be 2 map units. Okay? Alright, so that's 2 and 35. So that's what you're going to go ahead and do with the other problems. You're going to use the recombinant frequency to figure out how far apart are they. Are they on the same? Are these genes on the same chromosome, or are they on totally different chromosomes? Are they linked or unlinked? And then you're going to be able to use that information to map out where the genes are on the chromosome.